Hey everybody, welcome back to our monthly journal club meeting. This is Oliver Medvedik presenting to you from my bedroom library in the city of New York. Things are slowly opening up here. I think lockdowns are happening still across the world, up and down. You know, we're still in the midst of this pandemic, so trying times for everyone. So this paper is, even though we focus on aging, this paper is a little bit apropos. It's an Alzheimer's disease paper, but there's a little bit of immunology in this paper. So we will unpack this as we always do with all of our Journal Club articles here in this session. So once again, this session is brought to you by Leaf Life Extension Advocacy Foundation, and thanks to all of our heroes and other supporters for making this all happen. So let's, without much further ado, let's kind of, let me share my screen. I'm going to flash up the paper that we're going to discuss. And this is a paper that just came out actually this month. It's a Nature paper. And I'll give you a little background on it as well. So share this right here. So the title is The Innate Immunity Protein IFIT-M3 or IFIT-M3, I'm not exactly sure how to pronounce it, modulates gamma secretase in Alzheimer's disease. Okay, so it's a very compact, self-explanatory title. But basically, so let me unpack that a little bit further. So what is IFIT-M3 and how does it modulate gamma secretase in Alzheimer's disease? Well, that's the point of this paper. It is basically a protein that's activated by the innate immune response. So just a little quick background. You know that we have an acquired and an innate immune response in our bodies. So the acquired immune response is antibodies. So something that, you know, if if we're unlucky and if we are lucky and we had COVID-19 and it left our bodies, then we have an antibody response that's floating around to hopefully, if the similar strain infects us, it'll kick it out with a more robust response, and you know we can bounce back much quicker later. And that's the acquired immune response or learned immune response. The innate immune response is basically a whole bunch of different proteins and other systems in your body that kind of are built in, baked in, that will recognize very common factors in many infectious agents and will kind of thwart them. For example, our tears have enzymes like lysozyme that chew up bacterial membranes or cell walls and break them apart. So that's the innate immune response. So there's a protein that is upregulated called IFIT-M3 in response to infections. And this paper here shows that it modulates this enzyme called gamma secretase, which is kind of a key enzyme in Alzheimer's disease. And I'm going to stop sharing here. And the important part of this paper is this is the first paper, I believe, that was published that basically gives a kind of a molecular link regarding this protein, this IFIT-M3, which interacts with gamma secretase and modulates its activity. So there's this connection, this molecular connection between the immune response, specifically the innate immune response, and Alzheimer's disease. And one other reason why this is kind of crucial and might be really important is if you've been kind of reading the literature about Alzheimer's disease, nobody really knows, what causes Alzheimer's disease. And I'm going to go through some little review slides here, but it's the major causative disease behind dementia. So dementias basically are clinical manifestations, that are measured by memory loss and delusional behavior and progressing, to eventual death. And Alzheimer's disease, accounts for about 60 to 80% of causes of dementia. So it's pretty, it's something that's very severe, and 1 out of 3 seniors is going to have a dementia that may be caused by Alzheimer's, and that's a very shocking statistic. And just to throw out another number here, 1 out of 6 deaths in the United States is caused by Alzheimer's disease. So that's, if you think about it, you think other things might get you, that might be accidents, but no, it's actually Alzheimer's disease, and those deaths are probably an undercount because of a lot of times people unfortunately die of this disease through indirect means, because they trip and fall, and they have other issues as well. So it's a huge problem. So anything that we can do to try to crack it is, and it affects, you know, all of us. We you know, is almost 1 degree of separation for all of us, you know, or 2 degrees of separation. You know, a lot of us have seen relatives that have been suffering this disease. So anyway, anything that we can do and learn about this disease is vital in the literal and figurative sense to basically cure it. So this paper addresses one facet of this disease. And again, we don't really know what are all the triggers for it. And the interesting thing is that there might be a lot of triggers. So there's certainly genetic factors that elevate the risk. So ApoE4, which is a protein, you know, which is it's a mutation in a protein that's involved in cholesterol metabolism. That's a big one that increases your risk for Alzheimer's. There's also mutations in the gamma secretase subunits, which I'll go over in a moment. So there's hereditary components, but there's also environmental components. So certainly bad diet, smoking to excess, you know, having type 2 diabetes is another risk factor, concussions, for example. But there's also this interesting angle where there's been papers implicating the gut microbiome or defective gut microbiome, and also viral infections, herpes virus being one of them. Interestingly, also poor oral hygiene, which seems weird, but if you think about it, you have a lot of neurons in your, you know, in your teeth. And so, you know, there's this connection, which just seems a little bit out of left field, where, okay, well, now you have microbes that might, you know, trigger Alzheimer's. So what's going on? It's so multifactorial. It's almost like a you know, like an out of control octopus. How do you know what's actually causing it? And this paper actually kind of, I think, illuminates this facet of, you know, the implication of how the innate immune system might be implicated in the progression of Alzheimer's disease. So in essence, well, let's just go back to, let's do a little review here. I'm gonna go to this slideshow here. I'll put together from current slide. Okay, whoops. So this is a ribbon diagram of a gamma secretase complex. There's probably other ones out there, but this is the one I found online from a Wikipedia page, and I just annotated it by throwing. So in the paper, you see it's got all these subunits: NCT, APH1, which is in green, PS1, which is presenilin 1. PEN2, PS1, I think is orange. Yeah, red is an NCT. So it's a transmembrane protein, and it's a gamma secretase. So it basically, what it does, it's found in lots of cells, found primarily and also in neurons, and it cleaves this protein called APP, amyloid precursor protein. And there's other secretases as well that could be found mutated in other kind of versions of Alzheimer's. So gamma secretase is on the right. So this other transmembrane protein, which is basically bobbing around in the membrane, gets cleaved by these gamma secretases. And when it gets cleaved, it basically forms these little pieces called amyloid beta, which are found in different sizes, right? And as they basically get cleaved, it's posited that they do a lot of things. So this is just basically me plagiarizing from a review paper that's down below, called Oligomerization and Conformational Change Turn Monomeric Beta Amyloid and Tau Proteins Toxic. So we'll just ignore tau proteins from now. So what do these things do normally? It's proposed that, you know, there's a lot of papers that suggest they do positive things if you have low levels of them. So I highlighted one, which is kind of the focus of this paper, which these amyloid beta proteins might have antimicrobial activity. They might actually directly attack microbes. They also might help basically regulate synaptic function by activating acetylcholine receptors. So they do a lot of, you know, normal things. But if the level of these amyloid beta, proteins which are secreted gets too high, they can oligomerize, or they clump together, and that's where problems happen. So, you know, again, this is a kind of a trend in biology. You need to have homeostasis. Too little is too bad. You know, if you get rid of amyloid, APP, amyloid precursor protein, or gamma secretase, that's that's bad for neurons, that's lethal. But if you have too much of this amyloid beta, then that's a problem. These amyloid betas tend to clump together if there's too many of them and they can't be cleared out. So this is the amyloid hypothesis in Alzheimer's disease that goes back probably until the early 90s or maybe late 1980s, which posits that, you have abnormal cleavage, maybe too much cleavage, or, of APP. You have an increase in this, accumulation of this oligomerization of these, of these amyloid betas, which, here's a little diagram on the right, which causes them to clump together. And then they form these plaques, right? So, and these plaques do a lot of things. They can damage synapses themselves, basically kind of gum up the works. They lead to the phosphorylation of this protein that's inside of the cells called tau, which is a microtubule associated protein that causes these other things called neurofibrillary tangles to occur, which all of this then leads to neurotoxicity because now cells can't communicate because you need microtubules to transport things up and down the axons. These plaques can activate microglial cells, which can basically sort of like a positive feedback loop ramp up, destructive inflammation. So the inflammatory response kicks in. And all of this stuff, accumulates to essentially slowly build up to cognitive dysfunction and dementias. So there's all these different toxic effects. So this is from a different paper. I can get the link later, but you can see all these different things happen. But basically, the eventual goal, the eventual end point is cell death, neuronal toxicity, right? So this is just a screen grab of, plaques that are occurring, these little dark things that are amyloid betas on the outside, and then you have neurofibrillary tangles that are on the inside of cells. And so that's the, and eventually you get cortical atrophy. So basically your brain basically shrinks because you have a die-off of neurons, and things get really bad. So that's just a, a rapid, description of the clinical progression of Alzheimer's disease starting at the molecular level. And there's some controversy about this amyloid beta, hypothesis, whether or not the plaques themselves are the triggering event to form, hyperphosphorylated tau, or whether it's the other way around. But that seems to be the working model that a lot of scientists have agreed upon. That's, that's the most data that suggests that this is probably the most likely progression at the molecular level. So okay, so where does that lead us with this funny named protein, this IFIT-M3? Well, in that picture that I showed with the gamma secretase, it doesn't just exist by itself as a complex. There's other proteins that bind to it and modulate its activity. So basically, if you get, if gamma secretase activity needs to be upregulated or downregulated, there's other proteins that kind of pair up with it and modify its activity. I think there's other ones that, in response to different environmental cues, such as hypoxia, for example. And that will regulate the formation of amyloid beta peptides. So IFIT-M3 was discovered to interact with gamma secretase in this paper. So let's go back to this paper. So IFIT-M3 is basically a gamma secretase modulating protein. And the way they discovered this is the way a lot of things are discovered in molecular biology, which is essentially through a screen. Now, this isn't, so IFIT-M3 is part of the gamma secretase complex. So we're going to go to figure 1. So this was done through a chemical screen. I won't go through the chemistry behind this. It's something called click chemistry. Essentially, there's these little molecules that can bind. So obviously, the sort of the background is you can design specific things to stick to other things very specifically. Classically what we know is antibodies. All right, you can make an antibody that can bind precisely to one other molecule, right? And then you can identify. In this case, it was this molecule called E2012, which is known to bind to PS1, which is part of the gamma secretase subunit. And they modified this little molecule called E212B-pine, which they can then attach other things to it, and like a fluorescent, I think a fluorescent molecule, and then see basically what else is stuck to it when they visualize it on a gel. So this is this figure B is basically a Western blot. And that just means that it's a gel that was probed then with antibodies against different proteins. And it was basically a sort of a pull-down assay. They wanted to see what would interact, what things using these small molecules that is known to interact with PS1, what else would interact with it? And they pulled out this thing called IFID-M3. So this E212 minus plus that's found in B, I think that's a competitive inhibitor. So this is the, if you add that, it'll block the binding of this E2012B pine. And then you don't get this PS1, you don't get a fluorescent interaction with this molecule. So on the left where you see the minus, you can see these two bands, very weak band for PS1, but then they pulled out the sharp band for IFID-M3. So this was basically, we'll stop sharing here. So a lot of cool discoveries are made in molecular biology by just doing a very kind of, well, it's called a screen. You screen for things and you see what interacts without any a priori, information as to what you would expect. And then you take it from there, right? So screens kind of get a short shrift sometimes because it's sort of considered to be a fishing expedition for unknown things. But a lot of the really cool things that have been discovered in molecular biology have come out as a result of novel screens that people have invented to essentially fish out interacting, biomolecules that interact with something else that was never previously found, before. So they set up a screen, and they were fortunate to find a hit. Okay, so this screen was done. I'm not going to go through kind of all of this because this basically just they look at they do other interactions with other molecules, E for example and C, to kind of confirm that they are indeed interacting. Just figure 1F for example is done in neurons. Just scroll down here to basically see in vivo whether or not these PS1 and IFID-M3 actually interact, and they do. So PLA is called a proximity ligation assay. I had to look that up, but it's an antibody-based assay that involves two antibodies and that are conjugated to a nucleic acid probe that's then amplified with PCR and then fluorescently labeled. So it's an interesting assay. But essentially, this this is blue is in DAPI, F is in F-actin, and PLA is basically the red is two very closely interacting molecules. And so if you have PS1 and IFID-M3, you could see that you get this pinkish or orangish coloration, which indicates that the antibodies are in very close proximity to one another, and that they are very close to one another. Basically touching each other within the cell. So this is, where is this? This is in mouse primary neurons. Okay. So, so using this pull down assay, they were able to find that in vitro, in a test tube, that these molecules, interacted when they, extracted them from cells, and that they are also interacting using this, this proximity ligation assay within cells themselves. So, so they're hanging around each other, they're sticking to each other. So it's not clear now, kind of the next steps. So this is sort of, like a classical molecular biology paper. Guilt by association, IFID-M3, they know now, they know that IFID-M3 is involved with the innate immune response. I'm just going to scroll up here and go through the, what is IFID? Oh yeah. Interferon-induced transmembrane protein 3. Okay, so interferon are these proteins that basically are activate cells in response to viral infection, right? So interferon. So if cells are infected, the interferons are basically released, and they promote an inflammatory response. So it's sort of danger, upregulate your defenses. So so IFID-M3 is induced by interferon. So it's already been known. So that's that's where the innate immune response comes from. Interferons are part of the innate immune response. Okay, so we have this guilt by association already now. That it's binding to IFID-M3. It's co-localizing with IFID-M3. at least in mouse primary neurons. And now they want to know, well, is there any kind of functionality taking place here? Is it doing anything with, with response to gamma secretase? And I mentioned that gamma secretase is involved in the cleavage of APP, amyloid precursor protein, right? And the cleavage products, come in different sizes, and the typical ones are, I think, 40 peptides or 42 peptides. So that's amyloid beta 40, amyloid beta 42. So they're going to be measuring here whether or not amyloid beta 40 and amyloid beta 42 peptide cleavage goes up or down in response to, messing around with the levels of IFID-M3. So in figure 2, what they do is they are now looking in human embryonic kidney cells that are expressing APP, amyloid precursor protein. And I think they're looking at another cell line, these U138 cells, which are, this is in figure C, which are human glioblastoma cells. So these are cells that are basically a brain tumor cell line. So these are human cells. And they're knocking down using small interfering RNA. So you can throw in this RNA that basically downregulates messenger RNA and suppresses IFID-M3. So interestingly, it doesn't appear that knocking down IFID-M3 has a role in knocking down, APP, NCT, PS1, which are those are 2 subunits of gamma secretase. Beta actin is just as a loading control, but their siRNA is working because you can see IFID-M3 is knocked down. And then they basically measure secreted amyloid beta levels. According to this, there's a modest decrease. So they have the p-values here. I don't know what the percentage is, somewhere in the, I want to say 20% or 15% knockdown, but it's somewhere in the text here. But basically, when you knock down your IFID-M3, their very first experiments are showing a modest decrease in amyloid beta variants 40 and amyloid beta 42. These are the ones that oligomerize, by the way, and cause plaques. So you get basically a modest decrease. So next, what they did was they knocked out IFID-M3 in a different cell line. So these U138 cells. So in this paper, the good thing about this paper is that they look at a lot of different cell lines, and we'll see when we get to later figures, they actually even look at tissues from patients that have late onset Alzheimer's disease, and also 5X FAD mice, which is familial Alzheimer's disease. So these are like the typical mutant mice that are used in transgenic mice used in Alzheimer's studies. So they're looking at all bunch of different cell types they're doing these assays in. So it's it's good that they don't focus on just one cell type. It's just it's a lot of, so one problem with having different cell types is I have to kind of remind myself which cell I'm looking at when I'm looking at these figures here. So these are U138 cells. So they basically knock out these, so this is just background information, that they're knocking out the cells, and they're knocking out, I believe, using CRISPR, because empty vector is basically a vector for guide RNA. And so essentially the cell line that's knocked out, if they, add in IFID-M3, I think it's from transient transfection, and they can get it back again. And EV is just your negative control. So they measure gamma secretase activity in these cell lines, just ignoring E here, not for any particular reason, but because they basically are using it to analyze a drug that I don't really significantly kind of affects the outcome of this experiment here. But they're looking at how it interacts with a certain drug. So I just want to skip over that because it sort of, in my opinion, detracts a little bit from sort of the message I want to convey here. But in this cell line here where they knock it out, they measure gamma secretase activity. So this is I believe an in vitro assay using and looking at whether the expression or the cleavage of amyloid precursor protein and the amount of amyloid beta 40 and amyloid beta 42 that's produced. And you can see here that through the basically the bar graphs here that the levels vary. So EV is so let's see, so If they add in extra IFID-M3, your gamma secretase activity goes up. If they knock it down, it goes down significantly. If they, add it back again in the knockout cells, it goes up. And according to this, these differences are significant. So they did the statistical analysis. So basically modulating IFID-M3 activity in these cells by, knocking it up and down and using also small interfering messenger RNA and knocking it down using CRISPR. You could see that the gamma secretase activity in these assays in several different cell lines is affected and affected negatively, right? So less IFID-M3, it appears to be a positive modulator for gamma secretase, right? More IFID-M3, gamma secretase activity goes up, right? So that's good. That's, they're slowly kind of building up the picture here for what it does. Okay, so figure 3. So now we're getting a little bit, now we're getting into merging into our field here, which is aging. So they're looking at the effects of aging and the expression of APP or PS1 on IFID-M3 and gamma secretase. So they're looking at mouse models, and they're looking at normally aged mice, and they're looking at mice that are also, and starting with figure 3E, which is in kind of dead center, they're also looking at mice that are these 5X FAD mice, which are your transgenic, kind of hyper, they produce a lot of amyloid beta plaques because they have 5 mutations in their gamma secretase complex, which basically causes a lot of accumulation of these plaques. Okay. So what they do is, so they're doing I believe is that a Western blot? Yes, protein levels of gamma secretase. So they're looking at membranes of 4 and 28-old mice, 28-month-old mice, so older mice, old mice. Let's see, 5 per age, n equals 4 for 28-month-old mice. So those are the numbers. And they're looking at the levels of proteins. So, okay, so if IFID-M3 modulates gamma secretase activity, and we know that the levels of Alzheimer's go up, the interesting thing is that these are normally aged mice. So I'm not, normally aged mice, I don't think they don't get Alzheimer's. So I don't know what's, it could be that these levels are going up because they're more prone to infections. But we'll take a look at the 5X FAD level. But certainly even in normally aged mice, so here is, female 4 and 28 months, 4 and 28 months, they're looking at NCT, PS1. And then you could see, even just by staring at this Western blot, you could see a darker blob, which the darker the band, you basically have more expression of protein. And this is just basically relative intensity. So IFID-M3 levels go up with older mice. And when they do these gamma secretase activity assays by measuring the levels of amyloid beta 40 and 42, this goes up as well, right? So that's pretty interesting. Those levels go up in mice that are normally aged. Let's take a look at figure C. One problem with this paper is that they use a lot of different chemicals that basically that are either inhibitory or stimulatory, and it's kind of hard to keep track of them. And they do a lot of different. I think they do a pull down assay in figure C. I got to remind myself here. Let's see. Okay, so solubilized membranes were captured. So I63BPL biotin, I believe it binds to presenilin 1, and then you can see with a pull down whether or not you have more of well, this is basically like the Western blot here, but here they're looking at the membrane, basically localization of IFID-M3 by this molecule that interacts with IFID-M3, and I think the levels go up. I'll have to go back to that, but I think they're basically doing the same thing. They're doing a pull-down assay with a biotinylated chemical compound that binds to PS1, and then they're able to visualize a pull-down. Okay. So let's see, figure D, what are they doing in figure D? Okay, so these are mice that have been knocked out for IFID-M3. So now they're looking at knockout mice, so Western blot analysis. And if they knock out IFID-M3 in these mice, gamma secretase activity, you know, it's they say it's statistically relevant. So you get a modest decrease in amyloid beta 40 and 42. So you get the opposite effect. So if you see levels go up, gamma secretase activity goes up. If you knock it down, gamma secretase activity goes down. Next, they look at these 5X FAD mice, which are kind of the workhorse mouse, transgenic mouse in the Alzheimer's research community. These mice have huge levels of, of amyloid beta proteins. You could see here, so these are 3-month-old mice, 12-month-old mice, wild type. These are Western blots, so amyloid precursor protein, NCT, PS1. NCT and PS1 are the components of gamma secretase. IFID-M3 interacts. APP is amyloid precursor protein. And these are several mice that are from 5X. These are wild type mice, so 3 months old, 12 months old. You can see APP levels go up, IFID-M3 levels go up, all of these levels go up. And when they look at relative intensity of IFID-M3, as the mice age, these levels go up. So, so there appears to be a correlation, for some reason, having these 5X FAD mutations, if you have hyperactive gamma secretase activity, there's a correlation with maybe it's not all due to IFID-M3. I'm not sure. But there is a correlation with more IFID-M3 with higher levels of, higher levels of other factors. So whether or not that's driving the 5X FAD, or there's other, because one thing to keep in mind, as I mentioned before, is that there's other modulator molecules that bind with gamma secretase, not just IFID-M3. Okay, let's go back to this. So figure F, what do they want to look at? Western blot analysis for PS1 and FTF. So this is the N-terminal fragment. So just a portion of PS1 and IFID-M3 in membranes in 5X FAD mice. So now they do knockouts. So what are they knocking out? Are they knocking out IFID-M3? I think they're knocking out IFID-M3. What's wrong? Yes, so these are 5X FAD mice that have been knocked out for IFID-M3. So this is just, this could have been maybe in the supplemental. This is just showing that it's been knocked out, so you don't have IFID-M3. And in these knockout mice, so they do the good thing is they do these experiments in both directions. You boost the levels, gamma secretase activity seems to go up. You knock down the levels, gamma secretase activity goes down. And even in these 5X FAD mice, right? So amyloid beta 40 and amyloid beta 42 go down. And this is a immunofluorescence micrograph of neurons in the cortex and hippocampus of these 5X FAD mice, and basically plaques per millimeter. So they basically stain this with thioflavin S, and these spots basically correlate to plaques, amyloid beta plaques. And you can see basically just qualitatively that the levels in cortex and hippocampus, which is where you see, all these plaques primarily accumulate, certainly go down. And this is just a quantification of the levels of plaques. So, okay, so far so good. They're doing mouse studies here, and they're showing, they've shown thus far that IFID-M3 interacts, localizes with gamma secretase. Not only does it localize, but it seems to modulate the gamma secretase activity, at least for the formation of amyloid beta 40 and 42. So you knock it down, the levels of AB 40 and 42 go down. Mice get older, IFID-M3 levels go up, amyloid beta plaques go up, and also gamma secretase activity goes up. So there appears to be a one-to-one correspondence taking place here. So now we go to kind of the more interesting study, which is human brains. So these are humans. So this is association of IFID-M3 with the gamma secretase complex in human brains. So, so they have a lot of patients here. So expression proton, so they look at temporal cortex from human controls and frontal cortex. So these are areas that get really affected by amyloid beta plaques. And certainly the hippocampus, which is involved in memory consolidation. So they have about 76 controls for controls that are not suffering from LOAD, which is, I believe, late onset Alzheimer's disease, and N equals 80 with patients suffering from LOAD. And then later they'll break that apart a little further. They'll distinguish between LOAD L and LOAD H, which is basically patients that have kind of high levels of amyloid beta plaques, and they'll call those low level, and LOAD H, I think, high level, just so they can more robustly correlate IFID activity. So anyway, so A and B are basically looking at expression levels, and I believe this is this RNA, messenger RNA expression levels. So these so-called violin graphs, I guess, are basically all these little dots here are patients, and it's sort of like a scatter plot. And you could see that where they cluster, because you have a lot of variability between patients. So they're looking at temporal cortex and frontal cortex. Control, 76 patients on the left, 80 patients on the right. And then they look at expression levels of IFID-M3 in these two regions of the brain. So these are brain samples. I believe they're probably biopsy samples and messenger RNA. So this is probably most likely quantitative PCR. And you could see, here they're putting little stars to show it's significantly different, but you could see that there's a trend towards, in patients with late onset Alzheimer's disease, the messenger RNA levels of IFID-M3 tend to trend upwards. And in the frontal cortex, it's even more pronounced, right? So you have this definite trend of more IFID-M3 happening, of higher expression levels in these regions. C is basically a Western blot, so looking at the actual protein levels. So they don't look at, I don't think they did Westerns on everybody here. So S1 are basically your people that are suffering from LOAD. C, 1, 2, 3, 4, 5 are your controls. And these are just expression levels. So you could see that there's a lot of variability even amongst patients. So this, if you could see some patients have low levels of IFID-M3, some patients have high levels of IFID-M3, but you could see that the trend is you have in general higher levels of IFID-M3 with patients that have LOAD versus the controls, right? So this is the relative intensities when they basically look at of these, a pool of these bands. You can see that the trend, again, both at the messenger RNA level and the protein level, you have increased levels of IFID-M3. And this effect becomes even more pronounced if you then look at these samples and you look at, let's see, LOAD L and LOAD H. So what they do is they then separate relative intensities, and they group these two groups as, LOAD L, which is low intensity, and high intensity. So, one group will have, they've separated, I think the goal was to take two groups that are shifted by, I think, 1 or maybe 2 orders of magnitude and expression or significance between L and H. So you can, if you look at these samples here up, you could see S5 and S6, tend to have low expression levels of IFID, but then there's some samples that have very high levels. So they wanted to see if they can separate these samples out in patients, will they be able to see a difference in amyloid beta gamma secretase activity, right? And indeed they did. So if they took, if they separated the samples and kind of teased them apart and said, well, let's look at the samples that have high levels of IFID-M3 versus low levels of IFID-M3, would we see a higher level of gamma secretase activity? And they probably did this. Maybe they did this gamma secretase activity with everything combined, and they didn't see much of an effect, which was strange. But then if they teased them apart and said, well, maybe this gamma secretase activity is more pronounced if we separate it out by the intensity. If we kind of zoom in on, on these patients that are, that are diagnosed as late onset Alzheimer's disease, maybe we can see an effect if within subpopulations of patients. And that's indeed what they see, is that there doesn't seem to be a significant increase in gamma secretase activity in load L patients, but you do see a significant increase if you take the patients that have really high levels of IFID-M3, and the gamma secretase activity is then visualized much more, well, significantly higher, right? So, so moving on to E, and this is where it now kind of starts to tie into the immune system, the innate immune system. They start treating cells with interferon gamma, which is basically a protein that induces all sorts of antiviral effects internally within the cell. And E, I think they're looking at, this is, I want to say it's a mouse, primary mouse neurons. So I'm not sure why they're switching to mouse neurons and not human neurons in this case, but they're using mouse neurons here. And you could see that if you add, so this is the concentration of interferon gamma, the more interferon gamma you add, 10 nanograms per mil, 100 nanograms per mil, IFID-M3 levels go up. So IFID-M3 levels are going up. And when they take the lysate from these cells that have been treated with interferon gamma levels, as the levels go up of IFID-M3, also the gamma secretase activity goes up as well. So that is interesting. Okay. So what else are they looking at here? So figure G, they're looking at gamma secretase activity for amyloid beta, cleavage from interferon gamma treated neuron membranes. So similar assay. So I'm going to kind of skip over G. Similar effects, gamma secretase activity correlates with basically higher levels of IFID-M3, which are upregulated by interferon gamma. Next, what they do, I think they look at human, yes, so now they look at human, they look at primary human astrocytes. So cells within the brain, so basically supporting cells, you know, within the brain that are been treated either with PBS, IL-6, or IL-1 beta. So these are, you know, these are cytokines, I believe, that are responsible for an inflammatory response in response to infections, right? So they look at, so figure I is Western blotting analysis for IFID-M3 and PS1, which is presenilin 1 in primary human astrocytes that have been treated with either a control PBS, which is just salt water, well, phosphate buffered saline technically, and IL-6 or IL-1 beta. So here is figure I. So PBS and IL-6, PBS and IL-1 beta, and then you basically have a Western blot, and then essentially you have, when they look at IFID-M3 expression levels, it's kind of hard to see here. It's a little darker. Definitely if you zoom in, let me see if I can zoom in here. Yeah, we could zoom in, right? So you could see that, I could see it by eye, and certainly the IL-6 and IL-1B look darker. I think if you basically do a number of these Western blots, you can then make a bar graph that's on your right and show that IFID-M3 levels go up. And as they go up, your gamma secretase activity also goes up. So very, very, very interesting. Okay. So I'm gonna kind of, so we have one more figure to go, but that figure will we'll come back to it. That figure is another chemical pull down assay that is showing where IFID-M3 basically binds to gamma secretase. Let's go back to it, share screen. Okay, so they used a number of different chemicals here. So this is this chemical called L458 that binds to, so what that's supposed to show is this, you know, this chemical binds to these little binding sites, these binding pockets in the active site of gamma secretase, which I believe is in PS1. And without kind of going too much into this here, because this is sort of, you know, illustrating where it's binding. But the figure E here is essentially a Western blot that illustrates the binding of IFID-M3 with what's that, with the N-terminal fragment of PS1. And what this basically shows is these blobs that you're looking at here in E. It's an assay that shows that basically when you use antibodies to pull down these two proteins that are sort of stuck together using this chemical called L458, that both of them are pulled down. So essentially they both kind of stick together and interact. And these bands that you see, these dark bands, show an interaction. And this leads the authors to conclude that, that you might have, this cartoon diagram in figure F, that this is, they're hypothesizing that this is how IFID-M3 basically docks and binds to kind of this two regions of presenilin 1, this N-terminal fragment and the C-terminal fragment. So this is basically just sort of a follow-up of kind of from figure 1. Sort of more specifically, once they want to illustrate where the binding might be taking place. So, where does this basically lead us as far as a model is concerned? There's still a lot more data that needs to be accumulated here. But the model is that you have immune inactivation, and we'll just jump into figure G, that if you have microbes or viruses coming in through a variety of different, portals, maybe through neurons, that are innervating your gut, maybe neurons that are in your mouth, maybe, neurons that are infected in a variety of different, locations. But somehow if you have an infection that is then gets past the blood-brain barrier, this can trigger various inflammatory conditions and a release of cytokines, which then cause IFID-M3 to be expressed, right? So either, you have interferons being produced in, response to an infection. It could be a number of different inflammatory conditions. So you have these, pro-inflammatory proteins that are produced, they're released, and IFID-M3 goes up. And as IFID-M3 levels go up, you have an increased cleavage of amyloid beta. And these amyloid beta particles at a low level, they might have antimicrobial effects. There's papers that suggest that these peptides can bind and sequester microbes and other infectious agents, but too much of it will essentially cause cascade, which leads to the amyloid beta hypothesis, which leads to Alzheimer's disease. So very intriguing, very interesting. It's kind of the first paper of its kind that makes this connection between the innate immune response and inflammation and Alzheimer's disease. So it's it would there's a lot of follow-up that could be done here, I think. I mean, the list can go on and on as follow-up papers. I mean, are IFID-M3 levels higher in people that have chronic viral infections, for example? And are those levels correlated with higher levels of Alzheimer's disease in those patients? Do some people, are some people protected, from these effects? Do they have mutations that cause their IFID-M3 levels to not go up? There's just a lot to unpack here. There's just a lot of follow-up, papers that can come out of, can basically come out of this paper, that could shed light on this process. And IFID-M3, are there other factors? Are there other proteins out there that can interact with other components of gamma secretase or other secretases that are implicated in Alzheimer's disease that have a link to the immune response, right? Indirectly through, by being upregulated by various inflammatory cytokines, for example. But this is the first of its kind, and it's interesting that I think this is really cool, and that if this is correct, it provides kind of a handle on this kind of unwieldy octopus that's you know, that's Alzheimer's, where you have a lot of different inputs coming in that can trigger the formation of amyloid beta plaques, and there's many different avenues. And some people, you know, and maybe the, you know, the central theme for all of these different, you know, triggers is the excessive formation of amyloid beta plaques and neurofibrillary tangles. But what exactly triggers the excessive formation varies from person to person and patient to patient. Either, you know, and that, and, you know, so you might have this kind of wild variance of the triggers, but all of these different triggers, you know, cause the same thing to happen, which is an overabundance of toxic proteins. And somebody said you have the cribriform plate hypothesis. Was that the one with the drainage of the yeah?
That's right, with the stent where they're proposing to drill the stent.
Yeah, and that sort of ties into the same, like that's still there's still the amyloid beta neurofibrillary tangle hypothesis, right? It's just it's basically okay. So it's sort of looking at it from the back end. All right, so let's say maybe there's certain people that, you know, that can tolerate huge increases of gamma secretase activity and amyloid betas, but they can clear them much more readily, right? So maybe the older you get, you know, it's certainly, there's papers that suggest your ability to clear them out atrophies, if you will. And there's certainly, you know, and certainly there could be a tie-in to, you know, to the inability of, you know, some people's, you know, of their vasculature to properly, you know, kind of flush out these amyloid beta particles as they accumulate. So yes, there's, you know, there's that cribriform plate hypothesis, and that ties into the I'm blanking here.
So it's Leucadia, isn't it? That's doing. I think I'm pronouncing it right. Leucadia. Is it Leucadia? Yeah, it's Leucadia. That's the company that's doing it.
Leucadia.
Yeah, but I mean, they think it's basically washing the when you sleep. It goes basically like when you're asleep, the central nervous system fluid goes around your brain and then drains out. So when you're asleep, you drain your brain, and because as you get older, and it goes through these little channels in the cribriform plate, and as you know, and it closes up over time because bone, well, it just gets out of whack, doesn't it? You know, the osteoclasts and osteoblasts, and it gets out of whack. And in the cribriform plate, instead of bone density tending to typically reduce with aging, they think it becomes more dense. And these delicate holes, which the central nervous system fluid drains through, they fill up. So their solution is simple. They think it's literally an issue of maintenance, and if they can make the holes bigger again, the fluid should drain from the brain and take the majority of problematic amyloids with it. Will it work? It seems a low-tech solution, but wouldn't it be amazing if something simple could alleviate it?
Yeah, so yeah, that ties into, you know, lymphatic drainage. So you've got your lymphatic system, which is essentially your circulatory system for your not only for your immune cells, but also, you know, for your interstitial fluid that kind of, and a lot of things get flushed out into the lymphatic system. So there could be, you know, we could see that the amyloid beta and accumulation is sort of like the nexus of sort of a lot of things that can go wrong, where, you know, you can get things going wrong by having too much gamma secretase activity at one end, which is stimulated by a whole bunch of different factors, both hereditary and environmental. And then you have, your body's defenses that can kind of buffer that. And one of them is drainage or clearance of amyloid beta plaques through the lymphatic system and also through microglial cells. We discussed a paper earlier with microglial cell activation using gamma stimulation. which is, you know, your microglial cells basically can essentially clear out. They could phagocytose plaques out. So, you know, your body has, you know, so things can go wrong, but then you have a defensive system. So if any of these two systems get out of whack, your defensive system collapses, or your, you know, let's say the offensive end goes up, at homeostasis is thrown out of whack, and your amyloid betas and neurofibrillary tangles get too high for the system to then be able to deal with them. And then you basically at that point have a downward spiral, which is quite brutal, you know, for all of us who've actually seen this in the flesh.
Yeah, I've certainly experienced that in my family with my nan, unfortunately, which was a very sad situation. But with all these different sources we were talking about, like, you know, like there's like the microbiome, the gut microbiome, the, you know, the oral hypothesis where the bacteria in the mouth is colonizing the brain. There's lots of different sort of potential sources, and which is probably also why so many different types of dementias and things are being identified, because they're all not quite the same. It's like Parkinson's. I was talking to you earlier today about that. They've identified 2 distinct types of Parkinson's based on the source of the bacteria. So the question really is, what we should be asking, in my view, is what is the best place to intervene? And as you said earlier, if they've all got that commonality of the amyloids, then would that not make sense? Rather than trying to play whack-a-mole with all the different things, would it not make sense to intervene where all these things come together?
Yeah. Well, I mean, we could probably be trying a multitude of approaches. I mean, because my you know, my close family member, my mother is afflicted with potentially Alzheimer's. Not a definitive diagnosis, but because you need a PET scan to do that. But let's say that's most probably the cause. And, you know, she's not diabetic. She doesn't smoke. She doesn't drink. So it's clearly all your typical, you know, environmental factors. I don't think she's had any concussions to the best of my knowledge. So that kind of goes off the table. We haven't had any kind of genetic test done, so I don't know if there's a hereditary component. I looked at my ApoE4 mutations, and they're fine, but there's certainly many other mutations that can happen in gamma secretases. So I haven't done a thorough genetic scan on myself or for her. So, you know, so that leads to, well, perhaps there's some sort of other factor that's causing it. And so it can get very confusing, you know, where it's not clear what the trigger is for an individual, you know, to be afflicted in such a way. But if the commonality is indeed amyloid beta plaques and neurofibrillary tangles, and you can promote their clearance using a multitude of different ways, that would be very cool. And, you know, unfortunately, the success rate for small molecule targets of amyloid beta plaques is abysmal. I gave a presentation a few weeks ago at a conference, and a very small conference at a medical center. And I read a review paper about the pipeline for drugs in 2019. And there's like 170 different drugs, and like roughly almost like 60% actually are targeting, you know, are using the amyloid beta hypothesis, or they're targeting either plaques or they're targeting neurofibrillary tangles. The rest of the drugs are kind of more for symptoms, you know, replacing depleted neurotransmitters, that kind of thing. The last drug there's only 4 or 5 drugs, and they only work on sort of improving the neurotransmitter levels. And they don't really target the root causes of Alzheimer's. And so there's only 4 or 5 of those drugs that are approved by the FDA. And the last one was approved in 2003. And out of this pipeline of about 170 odd drugs, there's only 1 drug that potentially might be effective in a clinical type, in a phase 3 trial. And that's, I'm probably garbling. It's an antibody against plaques, basically. It's called dacumab or something like that. And I think it's from Biogen. And and I think it has some nasty side effects at very high doses, but it's the only thing, right? So we really have to kind of look at lots of other avenues for targeting amyloid beta plaques. So I'm actually personally intrigued by non-pharmacological interventions as well, because there's certain things that we can potentially be doing, such as the work that came out over the years, suggesting that gamma stimulation, we don't have time to go over this, but basically we covered it in an earlier journal club, I believe, where you can boost microglial cell activity levels by basically getting your brain waves to synchronize and fire at 40 hertz. And the hypothesis there is that that's like a high level brain like state. So your cells are probably very metabolically active and probably very active. And if they're very active, they might be producing more I want to say more, oxidants, and they might need more assistance from microglial cells, and that sort of stimulates them to kind of, I don't want to say that this ties into the hormesis hypothesis. I might be the first one to actually say that out loud, but to me, it sort of smells like a kind of a hormesis type of event taking place, where you're getting hyperstimulation of the brain, and it's known, at least in a lot of patients, that that these gamma wave activity, which is indicative of higher cognitive functionality, goes down with people with Alzheimer's, or gets apparent. So if you can kind of get those levels back to normal, you might have a nice downstream effect where microglial cells could now be activated, and as a side effect, they start munching away at kind of amyloid beta plaques that have been accumulating. That's certainly a possibility. It's certainly a good avenue to kind of very robustly pursue, because worst case scenario, it doesn't work, right? Doesn't seem that there's going to be any negative repercussions to trying. We certainly don't have a very full pipeline, pharmaceutically, so to speak, of interventions that seem to be working all that well.
There's also some data for senolytics. I know that it's everywhere in the field, but there's even some data for that suggesting that the reduction of inflammation in the brain also helps the situation. So there's been some work in mice, of course, it's always mice, but yeah, it may be possible reducing inflammation allows microglial cells and the various other immune cells in the brain to effectively clear house better, because obviously inflammation causes the polarization of microglia and other immune cells to switch, and then they become pro-inflammatory instead of, I think it's M1 and M2. I think it flicks from a pro-inflammatory, a pro sort of rejuvenation and a soothing sort of almost like profile M1, and becomes a more sort of inflammatory, aggressive M1 type. And inflammation definitely causes that to switch and become, deregulated. So who knows? I mean, I think I can safely say that broadly reducing systemic inflammation is probably going to help on some level. Mm-hmm. I think that's fair to say, really. I mean, inflammation is generally, over the short term, it's useful, but chronic age-related systemic inflammation, it's never a good thing, is it?
Yeah.
So there's that approach. There's the one that I spoke to you about earlier with the GAIM or GAME. That's been funded for quite a few years now by the Michael J. Fox Foundation, and they're using. We talked about it earlier before we were on air, but they're using a bacteriophage to dissolve various amyloids, and it looks like they can. The ones they've got, they do a whole range of them, so not just for Alzheimer's, but obviously for Parkinson's.
How do they get it in there? Do they have to inject it past the blood-brain barrier? Because that's pretty big.
I would think so, yeah. I mean, I would guess probably intranasally. That would probably be the quickest route to your brain, right? I mean, it sounds pretty grim, but I mean, what's the alternative? So, but yeah, I mean, that's something that I would definitely recommend people check out if they're interested, and that's by a company called Proclara, and I think they're now in human phase ones, or I think they're very close to it. So, I'm really quite optimistic about GAIM or GAME for that, because it could potentially address Parkinson's, Alzheimer's, various related dementias, and things like, you know, like we were talking about CJF as well, and even ALS. It addresses all of the amyloids, which is interesting. So yeah, Proclara. I think they used to be called Neurophage, but yeah. I think definitely, as you say, we've got to try different approaches.
Yeah.
The crib reform plate, that is a beautiful low-tech solution as far as I'm concerned. And, you know, it's had good results with, I think, ferrets, they were saying. They've tested it in mice, and I think they've tested it in ferrets.
Yeah, I've got to check it. I've got to check it out, because the last time I saw the data was that 2 years ago now. Was when it was presented at our meeting in 2019. So I haven't followed up. So I'd like to definitely check out the research in that direction. But it's beginning to look like, you know, that it's sort of, you know, it's sort of familiar to like cancers, right? There's a lot of different things that can go wrong. The bottom line with every cancer is you have out of control, you know, you basically, it's, you know, you have cells that divide in an unregulated fashion, that can metastasize, that can basically promote formation of vascular tissue, but to get to that activity, there's many different routes to it, right? And there's and the treatment may vary between different patients. So it could be the same thing with a lot of dementias, and particularly even, you know, Alzheimer's, where, you know, you do have an increase of amyloid beta plaques, but maybe it's more effective to target some of these more upstream causes. You know, maybe some patients are hyper, you know, a hyperactive innate immune response, and maybe targeting that versus targeting the amyloid beta plaque, you know, would cause the system to balance itself more readily. I don't know. We can certainly try approaches. Certainly, the pharmaceutical approach to date has been hit amyloid beta plaques and neurofibrillary tangles as sort of the nexus. But maybe we need to think outwards a little bit and see if we can, you know, if there's a better response to suppressing amyloid beta plaques by kind of saying, well, maybe we can target them not directly, but by stimulating microglial cells or repressing the innate immune response, right? Getting the same, maybe that will be easier to accomplish, you know, and maybe that would vary between patient to patient, and being able to then tease apart what exactly is going wrong, kind of like at the cancer diagnosis level, you know, what exactly is hyperactive or not active in subsets of patients that is causing their amyloid beta load to go up, and maybe targeting that specific event and causing those levels to go down. I don't know, thinking out loud here, but certainly it's the statistics have been really not very promising with pharmaceuticals. I mean, they shouldn't stop trying, but you know, it's sort of depressing to look at the failure rate for small molecules and antibodies that have been, you know, used to directly hit amyloid beta, you know, itself. Maybe we need to kind of look out a little bit and say, well, maybe we can indirectly affect the amyloid beta levels and neurofibrillary tangles by hitting things that are kind of a little bit upstream, if you will.
Well, the gut microbiome is probably a good start. Things like low-tech solutions like fecal transplants or probiotics with the right sort of balance of healthy bacteria to seed the gut where it's become negative and taken over by a harmful bacteria. There's really quite good evidence, certainly for Parkinson's, can be reversed by dietary interventions in some cases. Obviously, it doesn't work across the board because, as we were discussing earlier, if that amyloid disease has got multiple sources, it's only going to work on the ones, you know, that you're targeting, like the gut microbiome. But it's worth investigating, and it may apply to Alzheimer's as well. It could be, if it's a gut microbiome issue, that's something that could be relatively easy addressed with fecal transplants. I know it's a bit grim, but yeah,
well,
they had success with fecal transplants for other conditions, obesity. I'm pretty sure they've addressed that.
Yeah, and like you mentioned, it, you know, if it might only work for a subset of people. If your trigger is that you're having a pro-inflammatory response because you have something wrong with your, you know, with your gut, that is basically leading to, you know, pro-inflammatory signals, you know, from your neurons from your gut, then perhaps, you know, that would work in this case. But if your problem is that you have amyloid beta plaques due to a concussion, that's not going to help probably.
So in conjunction with the different approaches, we need better diagnostics so we can work out which specific source it's coming from, and then approach it from a more sort of what I call it personalized medicine, or whatever, or individualized medicine approach, where, you know, that treatment is tailor-made for you. So if you've got Alzheimer's or you've got Parkinson's from your gut, then that's where you target. But if it comes from a concussion, as you say, or somewhere else, then you need to focus on that. So, and I think that's the way it's got to go, because we're all different and we all respond to different, you know, compounds and drugs in different ways, which is probably why they've not gained much traction with small molecules.
Yeah, and just to remind everybody, compounding all of this is basically the boogeyman that's behind all of us, which is aging. Because as all systems age, homeostasis can't keep up. You know, perhaps by having a very bad diet and having many infections and having a concussion, your immune response and other responses are perfectly capable of suppressing amyloid beta plaque, keeping up with elevated levels in your 20s. But once those systems basically start to falter, then homeostasis basically completely slides off the table, and you basically have an exponential increase at that point. So, aging is still the number one driver. You have all these triggers, but if your cells are still, are healthy enough and young enough to keep up with the damage, then that's fine. But eventually all those defensive systems are going to falter, and that's why aging is fundamentally the number one driver of Alzheimer's, and as well as other diseases of aging, if you will.
If aging is the driver, Oliver, we're going to obviously have to do something and get them out of the driver's seat and let us take over. So that's the solution, really. There's a lot of interesting stuff going on. I remain hopeful. We're starting to see more diversity in approaches now.
That's why I think things like non-pharmacological interventions, like this, what you mentioned with the cribriform plate hypothesis, the 40 hertz gamma stimulation. I'm optimistic that they might have an effect, but that's assuming that your cells are still young enough to be able to be stimulated by these interventions and be able to, even drugs against Alzheimer's that clear amyloid beta plaques. The assumption is that your microglia can bind to these antibodies and clear them up. But once your cells can't, start to lose their own functionality, you basically have the specter of aging that is going to cause these therapies themselves to falter at an advanced enough age. So these therapies I do think we should aggressively pursue these therapies, and I think they will definitely buy healthy years to everybody. But without actually tackling the root causes of aging, eventually all of these therapies will falter in the end.
Yeah. You can't, it's like slapping a band-aid on something, as Keith would say.
Yeah.
If you don't address why, the foundation of the problem, eventually the walls of the house, if you will, are going to fall down no matter what you do. You can slap as much, I think you guys call it spackle.
Yeah, plaster.
Like filler
spackle.
Or something.
Yeah.
Spackle. You can put as much spackle on the wall and in the holes in the wall as you like, but eventually everything's going to fall down, because if you don't sort the foundation out, you're in trouble. But we've got to keep on keeping on and try some different approaches. I know that the field was unfortunately dominated by a lot of senior elite scientists, as they called, who were sort of married to the amyloid hypothesis. It was tau or beta, and anybody else trying to get other approaches through were being actively blocked by, I'm not naming names because I can't say really, but we know that this happened in the industry, and there was a big scandal earlier this year where some, it all blew up, and basically it turned out that a lot of very senior researchers had been keeping the lid on younger people coming up in the field and preventing them from trying these new things. That's got to stop, really, and I wouldn't be surprised if it happens in cancer research and other fields where people at the top are actively stopping people who are new to the field coming up and trying new things, and it's really got to stop. I don't know how to fix that, but it's a problem.
Yeah, it's hard because it gets, we can table this discussion because it gets into human psychology and ego, right? So we can talk about what's the driver. Is it money? Is it power? Is it? Fundamentally, it gets.
What was it you called it? Monkey politics.
Monkey politics, primate politics.
Yeah, I remember that from a few years ago.
Yeah, so we're all guilty to some level of practicing primate politics. So more research, more good research, less shit slinging.
Basically.
Pardon the French. Yeah.
Oh dear. I didn't know monkeys could speak French. Oh well, never mind. Oh dear.
And I apologize to the French. That was I think I just started an international row right now.
Oh God. They want their statue back. But anyway, joking aside.
Yeah, the Statue of Liberty.
Yeah, they gave it you.
Well, that's a gift. You can't take that back. No,
I don't know. I don't know what the rules are. Oh well. Anyway, so that was interesting and opens another vista of possible approaches, and I guess more research needed. I'm always going to say that because that's what scientists always say at the end of the paper, don't they? Yeah,
but it's good. We're living in an interesting time where we're seeing a lot of data and loose threads get tied up and kind of consolidated. I'm optimistic that these are all going to connect, and they all seem to be connecting, and that we are slowly coming out of the woods, and it's not just basically laundry lists of things that go wrong and people are like, well, what the hell, there's like all these millions of disconnected facts, right? So it seems that these facts are all connecting, these observations, all this data is connecting and seems to be pointing towards one direction. I hope that in 5-10 years, I don't look back to what I'm saying now and everything just blows up and says, oh, we were completely deluded. Now we don't have no idea what's going on. It seems that that the data is consolidating in roughly the same direction. And I think this is the most optimistic I've been seeing this field, within the being embedded in the past 20 years. So By that I mean, aging and longevity, and it's amazing how far we've come from, a few decades ago with where the first genes in C. elegans were discovered that can extend the lifespan in a multicellular organism. It's amazing, and how far we've come since then.
We've come a long way. We'll get there.
Yep.
Hopefully we won't miss the boat there, and longevity for all, if you want it, of course. If you want it, no one's going to be forcing anybody to take anything they don't want to. So, but we'll get there.
And remember, it's not just about living longer; it's living healthier. If you don't want to live past a certain amount of time, that's fine. But nobody wants to live the last few decades of their life miserable and in pain, and having severe memory loss. I mean, even people who don't want to live beyond a certain age don't want that. I think we can agree on. I think we can all agree on that. Unless you're really masochistic, I don't think you want that.
No, I don't think anybody wants that.
Yeah.
So that's it. So keep on supporting the scientists out there who are doing wonderful work. Oliver is one of them, and there are many others as well in the field now. I'm glad to say. 10 years ago, people used to not take the field very seriously, but it sort of blossomed. Hopefully we'll have solutions in the next decade or two, maybe, but we don't know. We just have to keep on keeping at it.
Mm-hmm.
So there we go. So anyway, I hope everyone's enjoyed the paper, and I guess we'll be back next time.
Is that close to Halloween next time?
It is, yeah. So it might be a little bit of a spooky edition, as is our usual seasonal sort of style.
I believe that is October 27th on my calendar, Tuesday. It's the last Tuesday of October.
I think the Zoom's booked up for that. So, but
really?
we can probably do
it on Wednesday or something like that.
Or we can do it just an hour earlier, maybe.
Nope.
An hour or so earlier.
No, that might work for me too.
It'd be good if we could do it on Halloween, of course. That would be good.
That's a Saturday.
Oh, that's true. Yeah, it is, isn't it? Just noticed. Darn. Oh well, never mind.
Okay.
We'll bring a horror story called Aging anyway, and that'll set the scene for Halloween. So anyway, thank you very much to everybody who's joined us today, and to our monthly patrons, the Lifespan Heroes, for making this and the various other shows that we do. And we do lots and lots of things making all that possible. So thank you very much. And if you're interested in supporting us and getting more involved, do check out the heroes at lifespan.io/heroes and see if you maybe would like to support us. Everybody, but anyway, everybody else and included, we will see you next time, I guess.
Yeah, and I forgot one important thing to say when I introduced the paper. I forgot the authors and where this paper came from.
Oh, okay.
It was done at Memorial Sloan Kettering here in New York City. And let me just pull up my paper here. It's like I just jumped right into the paper because I was so excited about it. And I'm just going to quickly...
It's going to be off somebody's Christmas card list now for forgetting.
Oh, darn. Oops, let me just...
Honestly, such a faux pas among researchers. I've never seen before.
Yeah, and maybe we could splice this together later, so I can maybe introduce the. Oops. So the paper is by Ji-Yoon Her et al. And it is from both at the Weill Cornell School of Medicine and also at Memorial Sloan Kettering, I believe. There's a lot of different labs here, and also the Mount Sinai School of Medicine. Let me see.
Name.
Yes, it is. I wonder what takes place over there. Let me see. I want to make sure that I've got that right because I said, So Memorial Sloan Kettering. So again, her et al. Is the paper. The innate immunity protein IFID-M3 modulates gamma secretase in Alzheimer's disease from a multitude of labs here in New York City, Memorial Sloan Kettering Cancer Center, Weill Cornell School of Medicine, Rockefeller University, and Mount Sinai, as well as, I believe, Pfizer and the Feinberg School of Medicine, Northwestern University, which is in Chicago. I think that's, and I apologize for any other institution I haven't mentioned, but that is where the paper, that's where the paper's coming from. So totally forgot. Anyway.
I think it's not the first time you forgot, Oliver. I think we've been doing it for nearly 3 years, and I'm pretty sure I can't remember you always reading out the research. So I reckon you're off the hook.
Okay.
Oh dear. And on that note, I guess we will see you next time.
All right. Take care, everyone. Thank you.